next speaker, Josephine Chileshe from the University of Cape Town. Her topic is biomarker for Mycobacterium bovis infection in African rhinoceros. So Josephine is a postdoc research fellow in the Biomarkers research group at the University of Cape Town. The group's research interests are the expression of pharmaceutically relevant recombinant proteins in plants, insect cells, and mammalian cells, and the production of vaccine candidates for HPV, ASHV, and emerging infectious diseases, as well as a variety of diagnostics and vaccine for the One Health initiative. She holds a BS in Agriculture and Animal Health from NWU, MS in Veterinary Science from UP, and PhD in Molecular Biology. She's an animal health technician with a long-term research experience towards prevention and control strategy of bovine tuberculosis in cattle and African rhinoceros. She's skilled in comprehensive understanding and development of new vaccines and diagnostics tools necessary for disease screening and management strategy. Her research interest is in diagnostic of infectious disease for both animal and human health. However, her career aspirations include being involved in providing evidence-based scientific research on zoonotic disease for both human and animal health to help policymakers to resolve health sector challenges in Africa. Her other interests include Africa development, global health, teaching, mentorship, girl and women empowerment, and leadership. Let's welcome Josephine. Thank you.
Good afternoon, everyone. Can you hear me? Thank you so much, Mr. Opia, for that great introduction, and thank you so much for this opportunity. I hope everybody is okay. I'm going to, could you please? Okay, thank you. I'm just going to share some findings that we had to find during my PhD study, and this was really just based on developing diagnostic tools to diagnose M. bovis in African rhinoceros. African rhinoceros have economic and ecological value, but they are threatened by poaching and habitat loss, causing their population to reduce. In addition, infection with M. bovis, a causative agent of bovine tuberculosis, may be a threat to their conservation. Next, please. The death of free-ranging black rhinoceros in Kruger National Park due to bovine tuberculosis, published by Miller 2013, and cases in South Africa and around the world, emphasize the threat that this disease may have in disrupting conservation programs by restricting translocations. Since this finding, the KNP hasn't been able to translocate rhinoceros since 2016. Therefore, it is important that we have accurate and reliable diagnostic tools for the detection of M. bovis infection to ensure that uninfected animals are translocated. There are large knowledge gaps regarding M. bovis transmission, susceptibility to infection, progression of disease, prevalence, and different rhinoceros species in population, and the impact of rhinoceros ecology and conservation due to poaching. As you can see from the two maps, you can compare the historical distribution in orange and the current distribution in red. Therefore, to try and restore these fragmented populations, understanding aspects of M. bovis in rhinoceros requires reliable tools to diagnose individuals and to screen populations. Therefore, we propose to answer two main questions in the study. First of all, can M. bovis infection be detected in African rhinoceros? And also, are there any other cytokines that can be discovered apart from interferon gamma? So the immune response and diagnostic tests that are currently available, as you can see. So in generally accepted that cell-mediated response is predominant early in the disease, and later on humoral responses develop, which is measured by presence of antibodies. Which is measured by the presence of antibodies in serum, and cell-mediated immune response may gradually become weaker. The shift from cellular immune response to humoral to antibody production is associated with progression to clinical signs of M. bovis. Interferon gamma is a widely used marker of infection. However, other cytokines produced in M. bovis-specific immune response may be also useful for diagnosis for M. bovis. So the research questions included, what are the optimal ELISA? I'm really struggling with this. What are the optimal ELISA conditions for detecting African rhinoceros interferon gamma? Can a diagnostic AGRA be developed to develop M. bovis infection in rhinoceros? Can cytokine gene expression assays be used to distinguish between M. bovis infected and uninfected African rhinoceros? Thank you. Can release assays for cytokines other than interferon gamma be used to differentiate between M. bovis infected and uninfected African rhinoceros? In question 1, the whole blood from 5 rhinoceros was incubated overnight and stimulated with pocket nitrogen to be able to sensitize the immune system. And from the plasma, ELISAs were assayed and analyzed. Plasma samples were used for screening of three commercially available diagnostic tools and also 7. So as you can see in red, we have the antibody pairs that we used, and also in blue, those are the 3 commercially available kits for equine enzymes, should I say. So we use these proteins to be able to make do-it-yourself ELISAs to find out if we're able to detect interferon gamma in African rhinoceros blood. So on our right, we have the figure of our results. We show that the MapTech equine 3117 was the greater candidate, and we actually use it to further optimize for further diagnosis. And for the ELISA optimization, sample metrics, incubation temperature, linearity, reproducibility, and limit of quantification were used to make sure that this test is robust and we can carry on in the field. And in conclusion, for this part of the study, where equine interferon gamma antibodies can detect rhinoceros interferon gamma, and also the kit was successfully optimized for measuring rhinoceros interferon gamma, and the findings have been published already. So moving on to the next question, which was the interferon gamma release assay for the diagnosis of M. bovis. We followed a similar pattern to now make sure that the kit would be able to differentiate between infected and uninfected animals. Next, please. And the results showed that indeed this test was able to differentiate between infected and uninfected rhinos with a very significant level, as you can see on our left, and then on the right, we used an ROC curve to be able to calculate the cutoff for this test. And we calculated at 21 kilograms per ml with an area under the curve of 0.84, which is considered a very good test. An alternative cutoff value was also calculated because, we coming up with the first test, so we really wanted to just make sure that we had options, and we really wanted to take it into the field of Kruger National Park to be able to apply this to. So we really just wanted to be able to have not necessarily as many cutoffs as possible, but being able to have a leeway to be able to decide which animal gets translocated or not. So we were able to find that a cutoff of 61 and a cutoff of 21 picograms were the 2 cutoffs that we used. So if an animal is 21 picograms per ml, that means it is negative, it doesn't have M. bovis. But any animal that is above 61 picograms per ml was considered positive and was hence not translocated. Conclusions of this chapter. Conclusions of this part of the study were that the QF2 stimulation platform can be used with rhinoceros whole blood to stimulate interferon gamma ELISA production, and also the MapTech equine interferon gamma can be used to measure antigen-specific cytokine release in rhinos, and also the QFT AGRA could distinguish between infected and uninfected rhinoceros. And this work has also been published. So moving on to the last part of the study. Could you kindly? Moving on to the fourth part of the study, for question. We had to now find other cytokines that could be used as a biomarker in addition to interferon gamma, because the more panel you have, the more cytokines you have in a panel, the better the confidence in your test. So we used a gene expression assay for this to be able to identify the markers, and then from there we were able to find commercially available proteins for these genes as to be used in the ELISA. So that was our roadmap for the methods, and we used an RT profiler PCR array that is a multiplex gene expression, which had about 84 genes. So that's really a good number, and we're just able to just spread the net so wide to be able to just find any targets. Next, please. So the results showed that we were able to find about 6 in total candidates, and it was so interesting that interferon gamma that we had already developed was, but it's not on that list. Sorry. And then in the study, continuing on the results rather, we were able to now apply these targets into the population that was infected and uninfected to see if these markers will be able to differentiate between infected and uninfected. And we found that the CSL10 was able to give a significant difference between the two populations, and hence we went further to evaluate the protein of that gene, which is IP10. Next, please. The conclusion of that chapter includes rhinocytosine gene transcripts were successfully amplified. The use of multiplex gene assay has an advantage of screening a larger number of potential cytokine targets, and a gene expression assay may have a diagnostic potential. Next, please. And this work has also been published. Next, please. And for the evaluation of the IP10 as a biomarker, we were able to test just a few samples, and we were able to see that we compared the nil, which are not, this is the blood that is not stimulated, and the mitogen blood that is stimulated with mitogen. There was a difference, as you can see in our figure in the left, but the difference was that there was a spontaneous IP10 that was produced in the unstimulated blood, which may have issues with translating the results, because then if the unstimulated and stimulated can both elicit IP10, then what is really the point, or we won't be able to see the difference. So then we actually had to obviously do further analysis with that to be able to find a way that we can reduce that signal of IP10 in unstimulated samples. And then to our right, we're able to find a cutoff. We just went ahead and used what we had because the majority of the samples still gave us the good result that we wanted. So we're able to go ahead and calculate the cutoff. We were able to calculate a cutoff of 203 picograms per ml. As you can see in your right, that there was that difference between uninfected and infected rhinoceros. And when we ran the IP10 test with the IGRA test that we developed earlier on in the study, we were able to find that IP10 was able to identify an additional IGRA negative animal, which shows some promise that when you do use the combination of the two cytokines, you'll be able to detect more animals that may have or have bovis. Conclusions of this chapter, of this part of the study, included equine IP10 ELISA can detect IP10 in QFT stimulated whole blood plasma. Spontaneous production of IP10 highlights potential confounding effect of results, which was really the highlighted challenge for the study. And also IP10 has a moderate promise as diagnostic biomarker. We're not very confident about it, but it was able to give us some indication of promise. And lastly, the impact of the study in general was that we're able to use commercially available equine reagents to consider them for rhino samples, because as wildlife, I don't know if you know, but there's really lack of reagents available out there. And the interesting thing is hearing from my previous colleagues of actually they're able to develop reagents that really helps in the diagnostic sector. And also the first study to optimize a diagnostic IGRA for rhinos. And our study was also the first to describe a potential alternative biomarker to interferon gamma, and then also we're able to develop tools to make important advances for screening before movement. And I would like to acknowledge my PIs and group and sponsors and collaborators. Thank you very much.